Good morning, everyone. I'm Dr. Romana Mirde, and I welcome you all to this second MycoAsia workshop on the topic Microbial Genomics for the Beginners. I now ask Dr. Anandita Ghosh to introduce today's speaker, Dr. Gaurav Sharma. Anandita, it's up to you now.
Thank you, Dr. Romna. Very good morning to all of you. Uh, Dr. Sharma is an assistant professor in the Department of Biotechnology at the Indian Institute of Technology, Hyderabad. Earlier, he worked as a DST Inspire faculty at the Institute of Bioinformatics and Applied Biotechnology at Bangalore, and as a postdoctoral researcher at the University of California, Davis. He has PhD degree in microbial genomics from Jawaharlal Nehru University, New Delhi, while he was working with the Dr. Sri Krishna Subramaniam at CSIR Imtech, Chandigarh. The broad focus of his lab is on the microbial genomics, transcriptomics, and evolution to understand the physiology of new model organisms such as mycobacteria and vibrio. Uh, his lab is also interested in understanding the plant microbe interactions within Indian.
Okay, so thank you, Anandita. And
Thank you,
first
sir.
of all, I want to thank Dr. Shinoy for inviting me to this forum. It's again a pleasure to talk to so many young people and discuss, what we have been doing and what can be done by you. And obviously, if we get the opportunity to train a little bit in a way that allows you to understand and start something new or afresh, then it's always a great honor. So today I will be talking about microbial genomics for beginners. Again, this talk I will divide it into two parts. So first will be more into understanding the basics. And then I will discuss some of my work, which we have done in last 5, 6, 7, 10 years. And then we will just do some conclusion. Okay. So although I have been already introduced, but just to give you some, a detailed description. So I did my master's from JNU, then PhD from MTech, postdoc at UC Davis. Then I was a DST Inspire faculty at IBB Bangalore. And means at the time I started my lab, and then I'm a assistant professor from last 5 months here. And so my lab works on 4 major areas. First is microbial genomics and evolution. So we try to understand like the microbes, their physiology, how their diversity and their evolutionary patterns using this theme. And we are working with 3 major model organisms, or we have worked with 3: Myxobacteria, Vibrio, and SARS-CoV-2. So we also did some SARS-CoV work, although now we are not doing anything in that direction. The second is medicinal plant genomics and metagenomics. And here we are trying to understand that how medicinal plants might be interacting with the microbes. And based on, under this theme, we are trying to understand their microbiome, their chloroplast, their plant microbe interactions, to name a few. And then obviously we are collaborating a lot with several national and international collaborators. And another interesting theme which we have recently started, like from last 2-3 years, is that we are also making some prediction servers for biologists. So in case some experimental biologists want to use these kind of tools, they don't feel any trouble and they don't need to have any computational knowledge, and they can utilize these kind of tools. So again, we have not published anyone till now, but most of these, like these 3 tools, will be published this year, most probably. So, and we work on like several model organisms, but these are like just the few names. Like Myxobacteria is our main model organism on which we have been working. SARS-CoV-2 we work on. Vibrio we are working. Medicinal plants we are working, and as I said, like microbial communities, because we want to understand the microbial communities associated with medicinal plants. So we are also working with them. Okay. So this was just like a brief introduction, what we have been doing. And so let's go more into the topic of interest, of our interest, which is like what is microbial genomics. Okay, so microbes definitely are not new to anyone, I'm sure. Like everyone has heard these names, like kitanu, vishanu, and bacteria, small animals or small organisms. So all these names are very common names in our household, even, I must say. Okay, like if there is a small kid, and we will always keep saying, oh, don't play in soil, don't play in this area, you will get infection. So that infection is microbes, right? So microbes are associated with health and disease, with biotechnology potential, with an ecology and environment, with agriculture. So all the main prominent areas of our life, I'm sure there is an association of microbes with them. Okay, so if we talk about like broad, like very specifically, then pathogens, host pathogen interaction, sometimes novel drug targets, we can understand like novel biomarkers from microbes. Sometimes many of the antibiotics come from these microbes or fungi or some other uh, organisms. They can also help in biofilm production, bioprocess engineering, and even climate change. Microbes can also play an important role in that. And similarly, like to find out like new plant growth promoting bacteria, and maybe they can assist the plant to grow well. Okay, so basically, microbe or any kind of cell is a very integrated system to unveil microbial functions. Okay, now what is a broad framework of any kind of so right now I'm using the word omics analysis, but let's think about bioinformatics or computational biology as such. That what is a broad framework of any bioinformatics analysis or any computational analysis. So I'm not targeting experimental, because I'm assuming that you already have good exposure about experimental biology, where you try to understand like the function using mutants, using different type of gels, different type of western blots, northern blot, and so many different experimental evidences. But when we talk about computational work or bioinformatics, then we have to start from somewhere. Okay, so that somewhere is this point. And let me tell you here that this somewhere, sometimes it starts with your experiments, like your wet lab or experimental experiment, but sometimes you can directly start with someone else data. Okay, so and the good thing about bioinformatics or computation biology is that a lot of people have submitted their data which they have generated from experimental sources, and they have put it in an open access. Okay, so open access means that everything is like anyone can access that data. Okay, so like NCBI or ENCODE, GE or TCGA, these are like open access platforms. So if you are interested in your experiment only, then you can generate some kind of high throughput data or sequencing data. Okay, and if you are not interested in doing that, in going through that hassle, then you can directly work with some available data and dig in to understand some novel questions, okay, which others have not answered. Okay, so remember that for that, if you are taking this particular data set, then you have to study those papers which have used this data to understand that what kind of questions they have already answered and what kind of questions might be new. Okay, so once you have data, in majority of the chances, you will be working with the NGS data. Okay, sorry. So you will be working with NGS type of data. NGS is like next generation sequencing data, and this is like majority of the people will be working with this. Okay, then the second step comes with integration. So integration is like so remember right now you have data, but that data is always associated with metadata. So metadata is like so data is number of students. Let's say each one of there are 20 students. Each student is one type of data set, but metadata is information about each student. So what is the age of that student, what is the gender of that student, what is the family background of that student, what is the education qualification of that student. So the description about the data is known as metadata. Okay, so when you are downloading the data, you are also downloading the metadata, and then you are integrating that metadata with the data. Okay, and obviously you do quality measurement or maybe data transformation to bring it to comparable data set. Okay, once this thing is over, then we do like data modeling or analysis part. So this is more of analysis. Okay, and here we will do different type of statistical, machine learning, visualization, or any kind of analytic tools. And after that, we will come to some kind of conclusions, and we will try to understand the biology. Okay, remember the most important target of any kind of computational experiment is also understanding the biology. Okay, so like to a conclusion of this analysis will be some kind of biological understanding. Okay, so now let me just so now there can be different directions of bioinformatic analysis. So as you can, I'm sure many of you have heard these words like genomics, protein analysis, metabolomics, comparative genomics, transcriptomics. So what type of analysis we do in each of them? So in genomics, when we talk about genomics, we do especially genome sequencing, genome assembly, sequence annotations, gene finding or translation. Okay, if you are trying to understand protein, then sometimes we try to predict the structure, sometimes we try to, sorry, sometimes we try to understand the drug designing, that which kind of drug can be designed against this particular protein. Sometimes we do protein family assignment, that okay, what kind of this sequence belongs to which kind of family, or conserved motif, evolution. And if you have like multiple organisms to work on, then you can try to do comparative genomics. So again, comparative genomics you can do like within species, within different genus, within different family, order, or even like the whole bacteria or whole kingdom or animal kingdom. Okay, and when you are doing comparative genomics, you are majorly dealing with like either sequence similarity searches like BLAST, cluster, or you are comparing multiple genomes to understand them, or you are trying to understand the evolution or relationship between different organisms or proteins. Sometimes we also do metagenomics. So metagenomics and genomics are kind of similar, but the major difference is that the genomics we are doing with one particular organism in mind, and metagenomics we are doing for the whole community. So like any area, like I'm sure you know that every organism, like every organism cannot live alone. Okay, there are many bacteria associated with them, or many like maybe fungi also associated with them, or many small organisms also, like which can live inside or outside on their body, right? So that's why like people say that no organism is an island. Okay, so there are many organisms who are living all together and trying to affect each other either in a positive direction or a negative direction. So the only difference between genomics and metagenomics is that metagenomics is for the whole community, genomics is for one single culture or one single organism. Okay, so we can try to understand the microbial composition, we can try to understand host microbe interactions. In transcriptomics, we try to understand like gene expression patterns, or like which genes will get upregulated, which genes will get downregulated, and such kind of studies. And in metabolomics, we try to understand that what kind of metabolites or antibiotic sources any organism can be secreting. Okay, or what kind of interactions will be there in within small molecules or within different type of proteins or molecules in a biological system. But besides that also there are many minor but very important fields within bioinformatics, which is like drug designing, vaccine development, dairy technology, like how to increase the milk production, or how to increase the more protein content milk from an animal, or in forensics, like to understand like who is the culprit, who is the like who did something wrong. Crop improvement, like there are so many efforts going on in crop breeding, where they are trying to understand that how we can increase the production, how we can increase the resistance varieties. Okay, or how we can make like more proteinaceous or more fiber type of food. Again, designing enzymes for detergent, which we take it like very granted, but this is one big major area. Like every household is using like soap and detergent and washing powder and all these things. So constantly people are trying to understand and work on that which kind of enzyme will be better suitable for this type of detergent, which will not affect human health, but will remove all the stains on our clothes. And again, genetic counseling to understand like at the time of pregnancy or some pregnancy, it can be identified that if the new progeny will be carrying like some kind of disease or something. Okay, so Let's move into very specific field which we are targeting, which is genomics. So when we talk about genomics, can anyone tell me what is genomics? What can be defined as genomics? If anyone want to give your opinion.
Study of genomes.
Uh-huh. So, so basically, genomics is it's a branch of molecular biology. Remember this, that this is very important part, because we always think that okay, genomics or computational biology is just a separate branch of biology. It's not. It's a branch of molecular biology which is concerned with the structure, function, evolution, and mapping of genomes. So when we try to understand all these four aspects of genomes using computational biology, that is genomics. Okay. So again, genome, I'm using the bigger term here, genome, and genome also has DNA, RNA, and non-coding RNA, and non-coding DNA, and all those things. Okay, so now I'm sure you have heard another word, which is genetics, right? So can anyone tell me what is genetics? What is the major difference between genetics and genomics? Have you heard genetics word? Means you can
Genetics is
unmute
the study
yourself and
of
speak.
genes. Genetics is the study of genes, with genomes is the study of the whole genome.
Okay, to some extent. Okay, anyone else?
People are typing their answers
Okay.
rather than talking. But yeah, please speak up. You guys can speak up.
Yeah, feel free. Means I want to make it like more conversation type of. Okay, so yes, this is means also true that study of genes and heredity is genetics. But one major difference between genetics and genomics is that genomics is high throughput. Okay, genetics is not high throughput. Remember, do you understand the word high throughput? So high throughput is like you can study thousands and thousands of protein in a go. Okay. But in genetics, you will be studying one gene, or maybe an effect of two genes together, or maybe three, maybe maximum five altogether. But here in genomics, you can make it like extremely high throughput. Okay. So like these are just three very basic generalized difference, that here it is a targeted study. So this targeted study is the main part in genetics, which is so, guys, you can unmute yourself now. Okay. So, genetics is more targeted. Genomics is not targeted. Genomics is like, you are trying to understand everything. And here we are targeting only one or few genes, but here it will be like all the genes in the genome. As I said, that genomics is high throughput, global and high throughput, but genetics is like targeted and low throughput. And obviously, both of these are cleverly experiment designed, so I should not go here. But this thing is kind of same. That clever design part is required for both genetics and genomics. But here the problem or the advantage is that it has tons of data, and there is uncertainty because there is so much data, and computational power is required. And here it is like more experimental. Okay, so these are like some basic differences, and that's why, like, some people focus on genetics, some people focus on genomics, and some people try to bring both genetics and genomics together, because genetics is kind of a confirmation of any experimentation. Okay, if you want to understand, if you have like let's say if you have a genome, it has 1,000 genes. Okay, and I want to understand that which gene is responsible for motility. And based on these 1,000 genes, I found that okay, these 10 genes might be involved in motility function. So now, what can be done using genetics? That genetics people can take these 10 genes, do their mutant studies and other experimental experiments, and confirm that yes, each one of them, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, each one of them is involved in motility. So if you mutate this particular gene, the organism cannot move. Or if you mutate, so basically if you mutate one gene at a time out of all these 10 genes, the organism will not be able to move. So that is kind of a confirmation. This is not a confirmation. Remember that. Okay, I'm again saying this is not a confirmation. Genomics or bioinformatics or any kind of computational biology, it tells you that yes, this is a possibility. Okay, sometimes you can say 99% possibility, sometimes you can say 99.99% possibility, but for 100% confirmation you have to do experimentation. Okay, so the idea here is that using genomics or using computational biology, you can come up to some kind of broad conclusions, and later on experimental people can take those leads and confirm them that okay, yes, whatever this guy is saying is true. Okay, so now it's a big question, and I'm sure like many of you are here traditional biologists. So the main question here is that can a traditional biologist handle big data? Okay, or handle like this tons of data or high throughput data? Okay, so just to means you don't need to read as such, but the idea here is that mm, anyone can do an computational analysis first of all. Okay, obviously, if you have some kind of bioinformatics background, it will help you in doing that analysis with more precision. Okay, even if you don't have, there are so many platforms nowadays who can teach you or who can train you. Okay, like there are so many YouTube tutorials which you can follow. Okay, even such kind of courses like this or maybe earlier courses which Dr. Shino and MycoAsia team has organized, and the purpose of these tutorial is to help you a little bit in terms of understanding the basics of computational biology or some other field, so that you can learn, you can go home and learn it more enthusiastically. Okay, so yes, it is possible a traditional biologist can handle the data. The only prerequisite is that one must be prepared to battle through the initial confusion. There will be a lot of confusion initially. Okay, so it is not a very straightforward road. You will get confused, you will be demotivated that okay, I cannot do it. But the idea is if you keep doing it, if you keep working on those tutorials, if you keep practicing those tutorials, you can do it. Okay, and the only solution is that just start doing it and keep doing it. If you keep doing it, it will be done. Okay, so this is a very broad genomics roadmap, which according to me stand correctly, like at least in most of the cases. Okay, so there are three major areas where we usually work. One is like NGS, second is homology analysis, and third is comparative genomics. Okay, and each one of them has like several subparts. So such as like next generation sequencing will be further analyzed into genome assembly, genome annotation, genome statistics, and similarly homology can be sequence based homology, profile based homology. I will try to explain all of these like one by one, maybe not like in extensive detail, but at least give you some glimpse of it. And comparative genomics, when we talk about comparative genomics, we do like a lot of phylogenetic, a lot of synteny, and genome kind of studies, which help us in understanding the biological question. Okay, again, I will keep repeating this point again and again that the target is biology. We want to understand the biology. We are using tools or we are using computational tool to understand the biology. So there has to be a biological question beforehand, that what is the question. You do the analysis using tools, and then you get a biological answer that okay, yes, the question which we had here, it can be summarized, or its answer can be summarized in this. Okay, so we will try to understand these things, but let's first think about it. So have you seen a genome? How does a genome look like?
It's full of contigs, I would say.
Okay, what else?
Um,
So
introns and exons.
genome is basically ATGC only, right? It's very basic. So which I'm trying to say that genome is ATGC, a combination of ATGCs. Okay, and when we sequence it or when we assemble it or something, it's like this. Okay, so this is a 9 MB genome. So 9 MB means 9 0 0 0 0 0 0. Okay, so these many base pairs. Okay, so I hope you can see this. So this is a 9 MB genome, which means that there are 9 MB ATGC number characters. Okay, so no one can understand what it is, right? Like just by seeing this ATGC, you cannot say anything. Okay, so in basic words, we can just say it's a naked genome. It doesn't have any information. It's naked. Okay, now, so this format, like how I have represented it. So let's not see this, because this is just the information. So this is our genome from here till here and so on. Okay, till the 9 MB base pairs. Okay, so the first line is always like a description line, and after that it is the sequence, or sequence means ATG6. Okay, so this format is known as FASTA format. Okay, so any type of nucleotide or protein sequence, and so remember the word protein and nucleotide sequence should be written or must be written in FASTA format, and this is the standard FASTA format. So you have a description of any sequence, and you have a sequence. Okay, so this is an amino acid sequence, as you can see, and but it will be the same in both genome, nucleotide, protein, anywhere, and every description will start with this greater than sign. Okay, so the first character of the description line will be greater than sign. Okay, so I hope it is clear. If you even want to see such kind of denotation, you can see this. Okay, let me just quickly show you on NCBI page also. Okay, so this is a standard NCBI page. I hope you guys are able to see this. Okay, so as I said, that it's a complete genome, and if you see, so let me make it first the naked genome, how the naked genome looks like. Okay, so it will just take a little while, and so this is our genome, which is a combination of ATGC, and if you just keep scrolling till the end, it will be just ATGC. That's it. Okay, so there will be nothing but ATGC. Okay, I hope it makes sense, right? So now, question is, it doesn't depict anything. Okay, it just have a combination of ATGCs. Now, let me go back there, and this is in FASTA format. So now there can be different types of genome. Like when we see in the data, it can be of multiple types. I will explain this step later on, because I will explain first what is read. But so the idea is, when you assemble different type of reads, so reads comes from sequencer. Okay, so there is a sequencer like Illumina or PacBio, Roche, or Nanopore, or something like that. Even Sanger sequencer. Okay, it gives us reads, and when we assemble these reads, we get. So if these are my reads, so let's say these are my reads, they are overlapping with each other. So in that case, I'll get one read complement to this, one read complement to this whole segment, because they all are overlapping, and another read, another compilation of this. So basically, I got three big structures: 1, 2, 3. So these are known as contigs. Okay, so there are gaps in between which we were not able to fill, and that's why this type of assembly is known as contig level assembly or draft assembly. Okay, so these three assemblies are draft assembly. Draft means it's not complete. Okay, complete means if all the reads are covering each other and there are no gaps in between. Okay, so in that way, we will be able to make like one complete sequence without any gap, and this will be known as, and this will be known as a complete genome or a chromosome. Okay, so if it's a complete genome or a chromosome, it means that there are no gaps. But if there are gaps, then it will be at contig level. Sometimes we can put in between ends and make and call it like a scaffold. Okay, so the difference between contig and scaffold is scaffold are ordered. Contigs are not ordered. Okay. Okay. Yeah. Means I hope the writing part is a little bit tricky. So, so contigs are not ordered. Scaffolds are ordered. Okay, and scaffolds will have ends. Contigs will not have ends. Okay, so in that way, these are known as draft assemblies. This is known as complete assembly or a chromosome assembly. And that's what I was trying to say here, that this is a complete genome. It means there are no gaps in between, and that's why it's a complete genome. But if it's a draft genome, it will have multiple gaps. So in that case, it will be multiple contigs. Okay. So just to show you quickly. Okay, I hope you are able to see this. Okay, so let's say this is sequence 1, and then it will have ATGC. Okay, then we will have another sequence which will be another contig, as I said. So this will be sequence 2, another contig, similarly sequence 3, which will be another contig. So in this file, there will be, like, say, in this particular case, there are 3 contigs. Okay. And so I can show you like some example also, if that is fine. But yeah, means I hope you understood this. Okay, so there are contig level assembly and there are complete genome level assembly. Now, as I was talking earlier, that a nucleotide file will again look like this because it's a FASTA format, standard FASTA format. Okay, and similarly, a protein FASTA file will look like this. Again, it's standard FASTA format. One thing which you may ask is that here, if you see, this description line is in 3 lines. Is it right? So I told you guys here that description line has to be in 1 line only. It has to be in 1 line. But here, if you see, there are 3 lines. So is it right or wrong? Okay, let me just show you once again. Okay, so some editors, if you just want to, I just want to convey this part. So let's say this particular sequence name is sequence 1 only. Okay, it can, so description can be any of any length. Okay, it can be 10 characters, it can be 1,000 characters or something. Okay, but now if you see, I have made it like really long kind of description. But sometimes in editors, so I just want to pinpoint this point, that ideally it is 1 line only, but there are like this line, because it cannot be visible like in 1 line, that's why they made it like in 2nd line. But it's not 2nd line, it's still 1 line if you see here. Okay, so this is still 1 line. So I don't know if you have seen this. So this is in text editors, like any kind of text editor, not in Notepad, but in Notepad++ or EM editor or something like that, you will always have this kind of option of wrapping. Okay, so wrapping means that do you want to see each line in 1 line itself? If you don't want to see that, you can change this kind of format. Okay, so right now it is still 1 line, but visible in unwrapped wrapped format. Okay, so similar situation is here. This is 1 line and this is multiple lines. Okay, so this part can be multiple lines, but this first line, description line, will always be 1 line. Similarly here. Okay, so now this was a genome, but the question comes that how we can make this genome. Okay, because that's our target, right? Like if let's say you went to Goa and like you collected like some marine water, or maybe you collected some soil which looks interesting. Okay, and you want to understand that, okay, which kind of microbes might be living there. Okay, so now the standard procedure is that you take the sample, you have collected the sample using standard procedures. Now you culture them. Okay, so culture them, like put it on a battery plate, and maybe there will be so many colonies. Maybe you feel that, okay, this colony looks very interesting. Subculture them, make similar kind of colonies. Still there will be some contaminations. Again, take this particular single colony and make it pure culture. Okay, so this pure culture will have same kind of morphology, same kind of color, and no contamination. Okay, once you have this, what you can do, you can extract its DNA. Okay, extract its DNA and then use that DNA to do the sequencing. Okay, now you can utilize this DNA and do the sequencing. So once you do the sequencing, you will get the reads. Okay, so from each sequencer, the output of a sequencer is a read. Read means a sequence, but with quality information. Okay, we'll discuss this quickly. And then once you have reads, you can assemble the genome sequence. Now there can be another situation where you don't want to go through this subculturing step, or it's a possibility that many organisms which are living in that sample which you have collected, they are not culturable, and in that case you cannot culture them, but you want to understand everything. So what you can do, you can directly take the sample, soil sample or marine water sample or any kind of water sample, and directly do the DNA isolation from that sample. So whatever organisms will be there, they will be their DNA will be extracted. Okay. So once their DNA is extracted, again you go with the sequencing. So now remember, now you are not doing pure culture sequencing, right? So you are doing a community sequencing. So sequencing of the whole community. So then in that case, it will be known as metagenomics. So that's why they have used the term genome or metagenome sequencing. So genome in terms of if it is a pure culture, metagenome in terms of if it's a community, if it's the whole community, and again then do the whole assembly annotation. Okay. So this is a very standard procedure. Now let's quickly talk about like this sequencing, okay? Because if you don't know what is sequencing or how the sequencing is giving you the output, you will never be able to understand the further steps. Okay, so maybe first of all, I'm assuming that you all are aware, so I will just give you a quick glimpse of it. So there have the purpose of next generation sequencing, or the purpose of sequencing, is to sequence the organism or sequence the DNA, and so that we can understand that particular genome. Okay, now obviously sequencing was identified kind of in 1980s, and at that time people were just working with like Sanger sequencing. Okay, so people like Sanger and Maxim Gilbert, they discovered a novel technique to do the sequencing. Okay, so what they used to do like dideoxy DNTPs and try to then visualize them using electrophoresis. So basically do the PCR reaction using DDNTPs and then visualize using electrophoresis, and based on their band location, you identify that what will be the order of the sequence. Okay, but the limitation of this was that you cannot go more than 500 to 1000 base pair. Okay, so that is kind of a limit with this type of first generation sequencers. So then later on comes Illumina or 454 or Solexa or Iron Torrent kind of companies, which says that okay, what we can do, we can make this process more like parallelized. Okay, so we can run all the samples in a parallel fashion so that we will get like high throughput data. Okay, so that was their idea, and by parallelization of these sequencing reactions, they increase the total reads, total data size. But what they did interesting was that they wanted to do it, or they were not able to extend the length of each read. Okay, so each read was like 50 to 500 base pairs only, but they were getting like in the size wise, they were getting it extensively. Okay, then in 2010 or around 2008, these two new companies came into picture. One was PacBio and Oxford Nanopore. So remember, even here, DNA polymerase is working. Okay, so the idea is that DNA polymerase cannot do more than like 500, or then it will fall from the template, and that's why it cannot go like more than 500. Or even if it is going more than 500, it will start doing more type of errors. Okay, so that's why it was not able to go more than 500 or 1000 using in first and second generation. So now in the third generation, or PacBio or Nanopore, what they did, they said that okay, let's make the polymerase fix, and then the DNA will move through the polymerase. Okay, instead of polymerase moving through the DNA will move through the polymerase. Okay, in that case, at least it will not fall as such. Maybe it will not fall, and it will. And now using these two techniques, you can get like tens of kb, 10 kb or 50 kb or sometimes 100 kb length of read. Okay, on average. Okay, so there some reads will be very long, some reads will be very short. Okay, but in between, the average number will stay like around tens of kbs. So now it is more real time in nature. Okay, so like you are not doing amplification, which you are doing in second generation or in Illumina or Illumina or 454 type of scenario. So that's why it is not sequencing by synthesis. It is okay. So these two were more of sequencing by synthesis. Okay, you are synthesizing the whole sequence or multiplying it like extensively. Okay, so that's why these first and second generation are known as short read sequencing, and the third generation is known as long read sequencing. Okay, I hope things are clear here. Just to give you like more differences or more specialized information about each of these sequencing. So as I said, this is more sequencing by synthesis. This is also sequencing by synthesis. It is not sequencing by synthesis. And in first generation, they were using like DDATPs with different fluorescence, and they were detecting the fluorescence. And the whole thing was that relatively small amount of data was generated. And similar in second generation, they were amplifying all the templates during sequencing, and it was very accurate because the length they have kept very small, like 100 or 150 or 200 base pairs, and very accurate. So very accurate, like more than 99.9% accurate. Okay. In third generation, they work with like single molecule template, so that template will directly go and uh, walk through the polymerase, and then it will have low accuracy and it will have long reads. Okay, so so they are not mm, they were not considering the accuracy as a problem earlier, but later on they realized that accuracy is a major problem. So now let's say PacBio has come up with a different strategy where they are giving very high fidelity reads, which is known as like hi-fi reads. So they have a separate chemistry behind that, where the accuracy is also like equal to second generation. Obviously right now it is very costly, but like accuracy wise and length wise, now PacBio is giving the largest number of reads, sorry, largest length of reads and good accuracy. Okay, so this is just a quick example, like, so basically you can sequence the whole genome, you can sequence a part of the genome. Let's say 16s is the most amplified or PCR kind of molecule, because whatever kind of bacteria or microbes you might be working with, you will always be sequencing the 16s RNA using 27F and 1492 primers. So based on these, you can either go with first generation, like which is Sanger sequencing, which we usually do in our lab. Okay, in second generation, you can do and use Illumina kind of setting and get the data. And similarly for third generation also, you can do either use nanopore or maybe PacBio and get the data. Okay, so now if you quickly see the difference between these different facilities. So if you see like Sanger, Illumina MySec, Minion is a nanopore version, and PacBio, you will realize that Minion is giving like the largest reads on average. Okay, Illumina is giving like very low, but Sanger is like 400 to 900 only, but with very good accuracy. Okay, so if you see accuracy wise, now PacBio has also improved a lot. Okay, so as I said, like, hi-fi reads are giving very high accuracy and very good length. And data wise, if you see, Illumina is giving like the largest amount of data. Even Nanopore is giving very large amount of data, but Nanopore needs a lot of lots of DNA sample. In that case, Illumina can work with very small amount and it can amplify it and make it large. Okay. Now, another thing here is that this is long read, like runtime also. So it runs like for very small amount of time. Illumina, because it is working with sequencing by synthesis, it needs like a lot of time. And similarly, because Minion, it takes like a lot of DNA, so it can keep working for more amount of time and keep giving you like real time data. Okay. Another important thing, which is price, because price is also associated with what we can do, right? So means compared to 2000, like when people started like doing more of NGS data analysis, nowadays sequencing is very cheap as compared to 2020, as compared to 2000 or 2005. Okay, so now the sequencing is very cheap, even the data wise, cost per human genome, if you keep talking about, at that time, like in 2001, it was like $100 million, but now it can be done within like less than $1,000. Okay, so which NGS strategy is better? It depends on, first of all, your uses, like what kind of use you have. If you just simply want to identify 16 RNA, that which kind of microbe I have isolated, obviously Sanger sequencing is always the best, because in that case it will be cheaper. You can just do for one sample very quickly. And, so Sanger sequencing will work. But if you want to sequence a whole organism, then maybe Illumina is good, because Illumina can give you like a lot of data. But sometimes now what people are doing, they are using like a combination of Illumina plus either MinION or PacBio. So you take Illumina data and you take PacBio data, and then you do the assembly. So that is kind of a best standard considered as of now. And we will discuss it, like what will be the advantage. Okay, so this thing is the first step, doing the next generation or getting the NGS type of data. Now once you have the data, remember the target of this study is to make the genome, right? So do the genome assembly. So the next stage is genome assembly. And now what we have to do, so we already have the NGS data. Now we have to assemble it into a genome. Okay, now this assembly, it has like four important aspects. So one is like quality control, second is streaming, filtering, and data, and third is sequence assembly, like the main step, and then assembly validation. Okay, so these are the four major steps which you have to follow, and only then you can do the genome assembly. Okay, so now we will walk. I will try to walk you through like all these steps one by one. So now if you see, first is quality control. So first we want to understand, when we get the data, that how is the quality of that data. Okay, like is it of good quality, so that we can work with this, or is it really bad? If it is bad quality, then you can go to your you can go to the respective company, or the sequencer, like sequencer means the same people, and tell them that, okay, my data is really bad and I cannot work with that. I have paid you enough money to do the good quality work, and so this is a problem at your end, and please deal with this. Okay, so that is like quality control, where we will try to understand all these things. Then even if you have good quality data, some data will be of bad quality. Okay, so remember, like, you have to take a call here, that if 1% is bad data, it's still good data. Okay, but if 70% is bad data, then it's not good data, right? So in that case, you have to think in that direction, and based on that, you decide that, okay, the out of the total data received, let me just proceed. Okay, let me just proceed, because it just have like some amount of bad data. I can remove it and work with the high quality data. Okay, and then we do the sequence assembly using different type of assemblers, and then we do the assembly validation, where we try to understand, which assembly is better. So you can try like multiple sequence assemblies here. You can try multiple sequence assembly strategies, and then compare all those assembly outputs and say that, okay, out of these five, this strategy is giving me the best data. Okay, now if you see the conversion part here, so here we got FASTQ file. I will tell you what is a FASTQ file, or let me just tell you the FASTQ file first. So what does a FASTQ file looks like? Okay, so FASTQ file is a such kind of file. Okay, maybe it's not clear. So such kind of file like this. Okay, so each FASTQ file will have only 4 lines. Like each FASTQ read. So FASTQ is a read file. Okay, so read file means each read will be in FASTQ format. Okay, and each read will be made up of 4 lines: 1, 2, 3, and 4. Okay, First line will be description line, as we discussed earlier, same in as in FASTA. But here it will always start with @ sign. Okay, so description line will always start with @ sign, and then it can have any type of description, whatever you want, or maybe basically it is sequencer information. But so it means in case you want to understand it more, you can you can read this, that what is this description file. So it will have like unique instrument name, run ID, flow cell ID, flow cell name, tile number, XY coordinate, all these things. Okay, but let's not go in there. But the idea is that first line is description line, which will always and always start with @ sign. Then there will be sequence, what it has sequenced. Okay, so this is our point of interest. The third line is just a line with plus sign, which is a separator of a second line and fourth line. And then we have fourth line. So if here there are 50 nucleotides incorporated, there will be 50 quality score on this line. Okay, so like each nucleotide code will have a respective score, respective quality score, and this quality score will be according to this table, as you can see. So if the quality symbol is IHG, like I is the highest quality, which is like 40. So 40 means that there are one error in 10 to the power 4. 1, 2, 3, 4. Okay, you can. So there are one error in one, like 10,000. If Fred score is like 30, it means there are one error in 1,000. Okay, so Fred score 30. And if there are Fred score 20, it means one error in 100. If it is 10, it means one error in 10. So basically, one error in 10 means if a sequencer is incorporating 10 nucleotides, there will be one error. Okay, here if a sequencer is incorporating 10,000, then there will be one error. Okay, so in that case, based on this order, if the quality is really low, then it will be like this apostrophe sign, or all these different signs as in order. Okay, so I hope it is clear. So now, as I was showing you here, that we have FASTQ file. Now, first step will make the quality control report. So quality control report will tell us that, okay, these are the all quality scales, and based on that, you can judge or take a call that, okay, this is a good quality file or this is a bad quality data. Now, in the second part, we will do trimming or filtering of the data. So as I said, like we will try to remove the bad quality data. So here we will take FASTQ file, and the output will also be FASTQ. Okay, but these FASTQ files will be cleaned files. Okay, clean file means high quality files. Okay, we will quickly discuss. And then by assembly, we will convert these FASTQ files into FASTA file. Okay, so once the assembly is done, we will get the genome, which will be in FASTA format, and FASTQ file will be used. Okay, and in the assembly validation part, we will take those config file or FASTA file, and we will make like assembly reports. And again, assembly reports will tell us that, okay, this assembly is better as compared to this assembly. Okay, I hope all these things are clear. Okay, so now this is our first second step. Okay, we have seen the document. Okay, we found that, okay, it has some good quality reads, majorly good quality reads, and some are low quality reads. And based on that, they will say that, okay, if you see, so this is like kind of a report which you will get, and you will say that, okay, before trimming, you have this type of data. Okay, so if you see here, this is Fred score. Okay, so quality score is also known as Fred score. So let's say this is 30, sorry, and this is 20. Okay, 20 Fred score. So basically here there are 1 by 1,000, 1 by 100, 1 by 100. So in within 100 nucleotide incorporated, there will be one error. 30, there will be one error in 10 to the power 3. Okay, so basically 30 is a good quality as compared to 20. 20 is a better quality as compared to 10. Okay, so now it depends on you what you want to use as a quality score. So if you say that, okay, I want to remove all the reads which are less than 30. So that's what they have done. So they have removed everything like less than 30 here also and here also. And if you see that now this data has like no read like below 30. Okay, like majority of the reads are now above 30. Okay, so in that way, the streaming or filtering of both forward and reverse read has worked. Okay, I hope it makes some sense. Okay, on the x-axis is length of each read, that okay, from where to where each reads are, and based on that you can read this read these kind of quality reports. Okay, so for trimming we use like Trimnomatic or FastP kind of tools. Okay, and to make this kind of report we use like FastQC. Okay, so FastQC helps in making such kind of report, and then after seeing these reports you can use Trimnomatic or FastP and and get the high quality reads. Okay, so once you have high quality reads, what is the next step? So you have you have the good quality reads. Now you want to assemble them. Okay, so a very basic strategy like which majority of these tools use is that when you isolate the DNA from a bacteria, so let's say this were your pure culture, and you extracted like the whole bacteria. Okay, so bacterial genome. So what you do, you instead of sequencing it directly, you have to chop it in between. Okay, so you have to fragment it. So you have to fragment it into like various regions, various small fragments, so that those fragments can be sequenced by the sequencer. Okay, so as I said that Illumina, let's say Illumina has a range of 100 to or maybe 50 to 250. Okay, so if you want to get all reads around 250 base pair, then you have to tell the Illumina sequencer that okay, I want all reads in 250 base pair configuration. Okay, so in that case, the fragmentation has to be done in 250 base pair configuration so that the sequencer can take those reads and do the amplification. Okay, so in that way, the first step is fragmentation, and after fragmentation, you will get the DNA sequencer reads, which after the sequencer has run through, and then once you have the reads, what you will do, you will try to align them one to each other. Okay, and find out that do they have like kind of overlapping areas. Okay, whatever the overlapping areas they have, based on those overlapping areas you can keep extending the sequence, and in the end either a contig, okay, or maybe a complete genome like this will be ready. Okay, so this is like a very standard procedure. Okay, so now, there are so many tools which can be used for genome assembly. Okay, so in case you have like, let's say, third generation sequencing reads, you can use third generation sequencing assembly tools. If you have like normal second generation assembly tool sequencing data, then you can use second generation de novo assembler. So let's say these are second generation sequencing assemblers like Abysses, Mega Hit, Soap de novo. Okay, or maybe these are third generation, which are like Falcon, Canoe, Smart de novo, Maycat, Fly, Shasta. Okay, so whatever is your requirement, based on that you can try to utilize a tool, and that tool will give you like some kind of final conclusion. Okay, so now as I said earlier that sometimes there will be like let's say if you have good amount of money to do the sequencing, so the best strategy is that you do short reads and you do long reads both. Okay, so the idea is if you only have short reads, there will be gaps in between. Okay, like let's say here is a gap or here is a gap. Okay, and here is another gap, something like this. Okay, so this is one gap, this is another, this is another. Okay, so in that way you will not get like a complete genome. Okay, so what is a better strategy? That you instead of taking like along with taking long reads, you can also take sorry, instead, along with taking short read, you can also take long reads. Okay. So let's say now you got long reads, something like this. Okay. So now what will be the situation that these long reads will extend, or they may extend from the gaps. So here was the gap, but this section is covered here in the long read. This was a gap, this section is covered here. This was a gap and this section is covered here. So in that case, you can extend long read and short reads all together and then get like a complete genome. Okay, so I hope it makes some sense. So in that case, that's what they are trying to show here, that when you use short reads and long reads, you can club them all together and make like large context or large chromosome, like as a complete chromosome. So in that case, it will be easier. It will be like, obviously it will cost a little bit more, but the idea is that it will be it is doable, and you will be able to get like a complete genome instead of just a draft genome. So I am assuming that you understood this part, that draft genome is of low quality, complete genome is of high quality. Okay. So, and then there are some kind of genome assemblers which can take hybrid reads like Masurka, which can take like both second generation read and third generation read and do the assembly. Okay. I hope this is clear. Okay. So now let me just go through two more topics, and then we will take like 2 minute or 5 minute question answers, and then we will keep working on for the next hour. Okay. So now, as I said, now we have a genome, which is a combination of ATGC. Either it can be a draft genome, it can be a complete genome. But now we have a genome. But still, that genome doesn't provide us any information, right? Because it's just a ATGC. So we need to get structures within that genome. We need to get like which is a gene, which is a gene which is making a protein, which is a non-coding RNA, which is a long non-coding RNA, or which is a exon, intron, and all these things. Okay, so now if you see, means I'm sure you are already aware about the eukaryotic gene structure and prokaryotic gene structure, right? So prokaryotes have a very basic and like easy to understand situation where a gene will start with a start codon and with a stop codon. Again, another will start with a start codon and with a stop codon. Okay, and either they may or may not be in a promoter operon situation. So if they are, then they will have the same promoter. If they don't have, then they will have different promoters. Okay, so in that way it is easier. But in eukaryotes, the situation is a little bit more tricky. They have upstream regulatory sequence, downstream regulatory sequence, in-frame, intron exon situation, different promoters, different proximal control elements, and all these things. So you have to take care of so many things. So remember, each of the word which they have written here, each one is like a limit, what is the right word? Like a thing which they have to search to find a gene. Okay, so if someone wants to predict a prokaryotic gene structure, they just have to find start codon, stop codon, and UTR, and either they are in frame or not. But here they have to identify which is an exon, which is an intron, where are the boundaries, do they have a poly signal sequence, do they have downstream regulatory sequence, upstream down regulatory sequence, promoter box, all these things. So like eukaryotic gene structure prediction is like more complicated as compared to prokaryotic gene structure prediction. Okay, and when we start looking into it, the idea is that first you predict genes, that okay, which will be the coding. Let me see if I have that slide or not. Okay, I don't have that. So the idea is you first predict a gene by identifying that where is the start codon, where is the stop codon, and do that start codon and stop codon are in frame. Okay, I hope you understand the frame part. So the frame is +3 frame. Okay, so if it is a start codon and this is stop codon, they should be in a break of 3. So 1, like 1, 2, 3, 1, 2, 3, and similarly 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, and 1, 2, and 3. So 3 will be the stop codon end. Okay, so if they are following this kind of pattern, it means they are in frame. If they are not following that pattern, then it means they are not in frame. And that stop codon cannot be a stop codon for that particular start codon. Okay, so once you have identified all these kind of predictions, then you will get a gene. Okay, once you have a gene, the function is not over because genome annotation has two steps. One is identification of a gene or genes or different structural elements. But along with that, it also has the function part. That what is the function of this particular gene or what is the function of that particular long non-coding RNA or something. Okay, so for that, what we have to do? We have to do a lot of homology studies. Okay, we have to understand that which kind of domain that particular gene or protein has, which kind of is it showing any homology with any known protein, is it does it have any kind of signal domain or any target domain which might be assisting, which might be helping that particular protein for a function. Okay, and so on and on. Okay, we try to identify orthologs, we try to do homology search against NR database to know that which kind of proteins are already there, and based on that we try to identify their domain, site, families, geoterms, pathways, reaction, that in which kind of pathways or reactions they will be involved, and in the end we will get a putative name. That okay, this protein should be named as something like this because it is involved in this kind of function and it might have this particular type of domain. Based on all these criteria, we will say that this is this particular protein, and this particular protein will be involved in something like this kind of function. Okay, so this is a standard strategy. Okay, once we have gone through all these things, then this naked genome will be more visible, something like this. So this is the annotation. So each one, it means that this particular gene is from positive strand, this particular gene is from negative strand, like green is positive, red is negative, and in between there will be so many other type of genes. And we will quickly have a look at it once again. So earlier I have shown you the naked genome kind of thing here, but if you convert this FASTA into GenBank full, okay, so GenBank full shows the annotated, like the annotated genome. So annotated genome will have information about each gene, each RNA, each tRNA, each non-coding RNA, everything one by one. So let's say this is for one of the gene. Okay, I hope you are able to see this. Maybe my browser is okay. So this is like first gene, which is being encoded from 87 to 1439. Okay, so this is the location in the genome. Okay, so the location of the genome, location of this gene in this genome is from 87 to 1439. Okay, it is, they have named it as DNA A protein. So DNA A protein, I'm sure many of you might be aware, it's a chromosome replication initiator protein, DNA A, and they have named it as a DNA A protein, but they also need a unique tag because DNA can be like in every organism there will be a DNA A protein, but they cannot keep calling every protein as a DNA. So here they will name it a unique name, which will be only applicable to this particular organism. So this is MXAN0001. Okay, so this number will only be in this organism. That's it. Okay, and then this gene, because this gene will make a coding DNA, it will also make a coding DNA sequence, CDS, and again it will be named as same. The locus tag will remain the same. They will say that how they identify this particular protein. So they are saying that they identified by match to protein family HMM this, match to protein family HMM this. So basically these two families belongs to chromosome replication initiator protein, and that's why they have named it as a DNA protein. Which kind of codon start they have used, which kind of translation table they have used, what is the product name, what is the protein ID, and what is the translation of this particular gene into amino acid. So this will be our protein sequence of this particular gene. Okay, so now if you maybe you are aware, but let me just show you. This is 87 to 1439. Okay, so this is the gene, this is the nucleotide sequence. So if I click on this gene here, I hope you are able to see the color difference here, right? So it is becoming a little bit orangish. So if I click here, it will open a small window here. Okay, you will just see, means right now my, as you can see, the mouse shape has changed, which means that this particular page is hanging. Okay, so it will come up here, as you can see. Okay, and you can even see the faster sequence of uh, this particular sequence. So again, it's taking some time. Okay, so what it has done, it has broken this whole genome into this particular segment, 87 to 1439. Okay, and now this is the gene sequence of that particular protein which we have recently identified. I also want to convey that you can do the same thing by yourself also. So here in the nucleotide page, you will see that do you want to see the whole sequence? If you want to see the whole sequence, you will see the whole sequence. Otherwise, you can select a region. You can say that, okay, I only want to see from 1 to 10, only 10 nucleotides. If you click update view, you will be able to see only 10 nucleotides. Make sense? Or you can say that, okay, I want to see only 10 to 12. Okay, you update the view, you will get 10 to 12 sequences. Okay, so in this way, this will be annotated, as I said. Okay, and the annotated file you can also see in graphic part. Okay, so if you see the graphic version, then the graphic version will be similar to what I was showing you earlier in the PPT itself. So it will create a visualization here. Okay, it will take some time. As you can see, it is moving here on the top. Okay, so this is like a kind of a graphic version. If I zoom it in a little bit, okay, so you will see that there are like these many, all each one of them is a gene. Okay, so if I make it like really small, you will see the same kind of pattern as you were seeing in the PPT. Okay, but if I zoom it, so it has zoom option here. You can see like each one of them like very clearly. Okay, so it is a little bit more zoom, but if I make it like smaller, a little bit more smaller. Okay, so now you can see that this is MXAN1120 protein. If you hover over it, it will give you like more detailed information about this particular protein, that what is the name of this particular protein. So this is a hypothetical protein, which means no function has been identified. If you look at this particular one, it is again hypothetical. Maybe this one will have some function. Let's hope. So this is a putative S-adenosyl L-methionine S-methyl transferase. Okay, so this is this type of protein. This is its function, and you can keep digging into this. Okay, if you want to know, if you know any particular protein, you can just type its name, DNA, right? We just identified. So it will say DNA. When you click on it, it will automatically open the DNA protein. So this is DNA protein. You can see this, right? So if you hover over it, you will find that, okay, this is the DNA protein of our interest, and you can proceed with it, like whatever you want. Okay. So that's how we do the annotation. Okay. So now, once we have done the annotation, so first of all, the annotation, there are several tools which you can use for annotation, like Glimmer, RNamer, tRNA-seq. Will do something different, and then based on everything, you will get the whole detail, that okay, this is these are the proteins, these are the genes, these are the uh, tRNAs or RNAs or something like that. Okay, once you are done with that, you will be able to make like reports like this, genome assembly and genome annotation reports. So in the genome assembly part, you will be talking about like what is the total length, how many contigs it has, what is the N50 value. So N50 value is like if there are 10 contigs, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, then how many of like how many if you sum the length of top contigs, and it becomes the length, the half of total length. Okay, so let's say we have 10, we have 9, we have 8, we have 7, we have 5, we have 3, we have 2. Okay, so let's say total number is 32. Okay, so wherever there will be 16 total, that will be our N50 value. So 10, 9, 19. So 19, it already become 19. So 9 is our N50 value. So which means that our assembly is really good, because in the top value only we are getting our N50 value. Okay, so higher the N50 value, better is the assembly. Okay, similarly, what is the GC content? What is the RNA-seq mapping ratio? This is not relevant. And what is the completeness of the genome? Okay, so there is this tool Busco, which can calculate like is it really complete, or how much more can be left. Okay, so it will say that okay, it is like 84.8% complete. So it means that still there are chances of improving this assembly. And similarly, you will do the genome annotation, identify genes, transcripts, or RNAs, tRNAs, CDS, GC content, or maybe the density of the genes, and based on that, you can understand all these things. Okay, now you can also create like this kind of a circular genome plot. Okay, so there are several tools like Circos, Brig, circular genome viewer, CGDV, circular return. Okay, which will make like these kind of plots. Again, like you need to have a little bit of bioinformatics knowledge here also. Okay, like similar to other things, but the idea is if you keep digging into it, you will definitely be able to do it. One of the tools, which is Brig, you don't need too much computational knowledge to understand Brig. Okay, it is a web server, it's a like downloadable web server kind of tool which you can utilize directly. Like even experimental people can utilize it. Okay, you have to study this, but you know how to use it, but it will be easy to implement. Okay, others are like more command line based on command line. Okay, so now you can mention whatever you want to show about that genome here. So let's say you are interested in antibiotic resistance genes. Okay, and you want to show that on this genome, where are my antibiotic resistant genes? So let's assume the red ones are antibiotic resistant genes. So you can highlight them that, okay, here is one antibiotic, another, or something like that. Okay, so you can mention or highlight them here so that people will understand it very easily, that okay, it has 40 antibiotic making genes or secondary metabolites, and these 40 are located in this circle. Okay, so you can make these kind of diagrams using. So diagrams are good. Diagrams should be good because in that way, or visualization should be good because in that way you can connect that diagram with your story. Okay, so with this, I think this is like the end of first section at least. So the idea here is you isolate the DNA. Okay, I mean, this is a very extensive paper, and I really want you to read this paper. If even if you don't have time, please read this paper. Okay, it's a very good paper, very easy to understand. Obviously, means I'm not saying that you will be able to do everything by reading this paper, but you will get so many ideas from this paper. Okay, 12 quick steps for genome assembly and annotation in the classroom. Okay, so please read this paper. Again, this figure is from this paper only, and they have mentioned every aspect of genome assembly and annotation in this paper. Okay, starting from DNA isolation. So means first DNA isolation, you have to decide that which kind of sequencing platform you want to use, then do the assembly, then do the polishing, refine it, make it like complete assembly in case you can, and in case you have money, do the annotation, use multiple annotation tools, merge those annotations together, and then do the visualizations using Circos or something like that. So that is kind of a wrap up for any NGS based genome assembly, annotation, statistics, and visualization. Okay, so this is a very standard protocol. Again, you will find all these details in this particular paper. Okay, so let me ask you, like, if you have any question till now.
There are some questions in the chat box. So, Anandita, can you please read those?
Yeah, sure. Sir, a question from Manohar. He is asking that is the assembled DNA sequence single standard?
Yes. So yes, it is single stranded. So we try to sequence like N to C.
Okay, one more question from him. In case of whole genome sequenced data, how can he visualize?
So visualization, see, the genome is like really huge, right? So you cannot visualize this like one to one. But if you want to see each and every nucleotide, then browsers like this are of very much help. So here, if you see here, like it will go till last nucleotide. Okay, so if you want to visualize it like this, it is good. You can do it like this. Otherwise, genome like circular genome plots like this can is also one another strategy.
Okay, thank you, sir. The Sheetal, she asking that does removing the poor quality sequences during the timing will lead to loss of sequence data?
Yes, so it will lead to loss of sequence data. But see, the idea here is that if something is bad quality, it will create some problems later on. So it's better to remove the data. So never compromise on quality part. Okay, compromise on less data, that is fine. But if you will if you will compromise on quality, things will not be great. Okay, so yes, strict no on compromising the quality. Okay, like a standard practice is that never use anything, like I'm saying never use any read below 20 Fred score, although 30 is preferred. 30 Fred score is the preferred value, but never ever use anything below 20. Below 20 is like a clear no.
Okay, sir, one more question from her. Why contigs do not ends and the scaffolds do have?
Say it again. Why cuttings contigs do
Do
not
contig
ends?
exactly.
Do
Do
not
not
ends.
ends and scaffolds do have.
Sheetal, what do you mean by this?
Uh, Sheetal, can you ask the question, sir? What is your
Why
exact
countries do
question?
not ends?
Hello?
Huh.
Hello,
Huh.
am I
Yes,
Yeah,
audible?
Sheetal.
yeah, you can audible.
Yes, my question is, you said that the contigs do not have ends. Actually, it is have. I have not written. So why contigs do not have ends and scaffolds do have ends?
Ends. Means yeah, both of them have ends. So let me just
You
go to
mentioned
that.
that, so
You will,
that's why
you
I
will be
asked.
able to. So if you see here, let me just. Okay. So contigs are like. So if you have multiple reads, okay. So this is one contig, this is one contig, this is one contig. So it will also have one end, right? They will have end to C, right? So there will be an end. Similarly, scaffold, if you even put like ends here in between, and it will become bigger with ends. So you are saying and, it's not and, it's n, n, like n. So n means because contigs, because scaffolds are ordered sequences. Ordered means, so how we do scaffolding usually. Most of the time there is a reference. Okay, so we try to map these reads on reference, and we say that okay, this contig and this context should be after each other, and there are 10 gaps. So instead of having a dedicated nucleotide here, we just put 10 ends and extend these contexts into one scaffold. So it's not end, it's n, like ends. I hope I'm clear.
I
N.
exactly. I hope Shital, you got your answer from sir. Two questions from Vruti. One is how do we determine the Fred score? And in case of fragmentation, how do you determine the base pair length, 200 base pair or 300 base pair?
Huh. So basically, we cannot determine Fred score directly. The sequencer has this facility which can directly understand that, okay, what is the confidence, or how confident was the sequencer in saying that it must be adenine. So that is Fred score. Okay, so based on that confidence value, the sequencing platforms decides that, okay, it should be good Fred score, it should be bad Fred score, something like that. Okay, so sometimes it is confusing for the sequencer also, that, okay, it cannot be it that, and that either it can be A or it can be T. So in that case, it gives it a low Fred score. Okay, in terms of fragmentation, so basically many of these fragmentation to many of like after fragmentation, either of any type, either mechanical fragmentation or restriction engine fragmentation, they select the bands that, okay, these are 300 base pair bands, so select them all. Okay, or if they are interested in 200, they select based on 200. Okay. And how will it affect the quality of data? No, it will not affect the quality of the data as such, because it's just that different platform use different type of insert size.
Okay, dear participants, if you have more questions, you can ask Dr. Gaurav Sharma, sir.
Okay, perfect. So let's move ahead. Okay.
Yeah.
So Anand, you're saying something?
No, sir, you can continue.
Okay. Sure. So it looks like that I will not be able to go into my research part, but most probably in the next half an hour, I will try to convey some of the tools with some of the other things. Okay. So let's move into the second part, which I discussed, which is homology. Okay. Homology is like the most important thing in bioinformatics. Okay, like 90% of the time, means I don't have any reference for this phrase which I'm using, but 90% of the research in bioinformatics is usually identifying the homology only. Okay, so whatever, let's say if you have a protein, the first thing which you will do is identify its homologs, that, okay, which kind of protein have the same kind of sequence, which kind of protein have the same type of structure, which kind of protein will have the same type of domain, same type of motif, something like that. Okay, so we are always playing with the homology. Okay, now the good thing is that there are so many databases, so many curated databases which are already there, which have a lot of information about different type of sequences which people have sequenced over the period of 10, 20, 30 years. Okay, so in that case, you can always compare with them and find out that, okay, this is the best homologue among the existing information. And if some protein is like really really unique, then they can also say that, okay, we were not able to find any homology. It means that this protein is like really unique, which no one has sequenced this time till now. But so when we talk about homologs, so homologs can be of three types. One is like or two types actually only. So one is just homology, another is orthologs, another is paralog. Okay, so a homolog is just a homolog, but sometimes if the homolog is present in it has a common ancestor, okay, and then it get deviated, so they becomes like a kind of ortholog. So if you see, there was a alpha chain gene, and this alpha chain become in frog, in chick, and in mouse, so these three will be orthologs. Okay, but if the same gene is being like have multiple homologs in the same organism. So let's say alpha and beta are kind of similar, but alpha and beta in mouse, they will be more of paralogs. Okay, or if it's a possibility that there are two alpha chain genes in a mouse, then they will be more known as paralogs. If okay, so I hope this thing is clear. Okay, now whenever we talk about homology, we there are different type of targets when we talk about homology. So a most common target is that we want to understand who are the closest organism, and how they which kind of domains might be conserved in that particular protein or in their homologs. So a usual standard is that you have a protein, you try to do homology search using BLAST. Once you will do BLAST, you will identify different type of homologs existing in any individual, whoever has been sequenced at that time. Okay, and based on that, you can take those sequences and do the alignment using different type of other tools like clustral, omega, muscle, or some these kind of tools. And once you are done with the alignment, you can predict models and you can make phylogenetic trees using mega or XML or beast or different phylogenetic tools. Okay, so this is a very standard flow. Okay, like how we usually work. Okay, now so obviously, I'm sure many of you have already heard about sequence alignment, but even when we are trying to identify the homology, we are trying to align the sequences and find out that which kind of sequences are homologous to each other. Okay, so sequence alignment is like kind of going on in the back end, and where we want to understand that which are the identical regions or similar regions between multiple sequences. And the idea is that if something is similar, then it means that it's a consequence of function or structure or evolutionary relationship. Okay, so that is the most important part, that if there are similarities, it means they are because of some relationship between those two. Okay, now the sequence alignment can be of two types, global and local. And global is like when they try to cover like the whole sequence, and local is like if we are trying to find like a very specific site. Okay, the overall purpose is to find the optimal score. Okay, so based on optimal score, we say that okay, this is a good alignment and this is not a good alignment. Okay, and these are like different tools which people usually use for alignment, like cluster omega, muscle, maft, MView, prank, t coffee. These are like some very commonly used sequence aligners, and many of these are already available on many of these are online available tools. Okay, so you don't need like any computational knowledge as such to run these tools. Obviously, to understand their result and everything, you need computational knowledge. That's a different story. Okay, but at least you can try to run them. Okay, try to run them in in your lab for your work or something. Okay, so now again, there are many differences between global alignment, but the most important one is that global alignment is suitable for aligning two closely related sequences. So if two sequences are closely related, you should go for global alignment. But if they are diverse sequences or distinctly related sequences, then local alignment is a better strategy. Okay. So now when we talk about local alignment, the most frequently used one is BLAST. Okay. So I'm sure many of you have already heard about BLAST, but BLAST again works based on alignment only. And alignment means we need a mathematical formula, right? So when we cannot just see the alignment and say that, okay, this is a good alignment and this is a bad alignment. We need score. We need some kind of numbers based on which we will say that, okay, this is a good score. So it means that this is a good alignment. And if this is a good alignment, then it will be a good homolog. Okay. So this score part is the most important part. Now, how to calculate the score? That is another point. So what people have done, people have generated scoring matrices. Okay. So researchers have developed like scoring matrices, which they use to identify the score. Okay. Now, if you see, I'm sure you know that there are several amino acids, and let's say there are several nucleotides also, right? So Nucleotides are only 4, so you cannot do a lot in there. But especially for amino acids, what they have done based on these three major biological factors, which is conservation, frequency, and evolution, they have designed it in a such way that that similar replacement, if there is a hydrophobic amino acid and it is replaced by a hydrophobic amino acid, it's fine. But if a hydrophobic amino acid get replaced by a hydrophilic amino acid, it's a big deal. So in that way, they have tried to make these matrices in such a manner that if something is same, it is always the best possible scenario. But if something is going in a haphazard manner, then they give it a negative score to make the alignment score negative, which means that okay, this is not a good score. Okay, so now these two are the most popular scoring matrices. Like one is PAM matrix and another is Blossom matrix. Blossom is the most commonly used one. Okay, because Blossom is most commonly used one because it works with more similar sequences. Okay, so more similar sequences, and in that case, the scoring matrices are more appropriate. Okay, so the idea is that if you are able to see that if the same amino acid, if the same amino acid is with the same amino acid, it is a good alignment. So they give the maximum score to the same amino acid. But if something is similar but not identical, okay, so in that way they give it a lower value. So like -1, like serine and threonine, they get like -1. So if serine gets replaced by threonine, they give it -1. But if serine get replaced by, let's say, histidine, so serine with histidine here, okay, so it will get a 0 number here. So serine with histidine will get 0 number. Okay, or like tyrosine, if it gets replaced by histidine, it will get -2. Okay, because it is very different type of amino acid which should not be there. Okay, or let's say glycine. Glycine is like the most simple type of amino acid, but if it get replaced uh, by by like tryptophan or phenylalanine, then they are getting like -3 or -4 values, right? So in that way, this is a strategy in which you can identify that okay, this is my alignment. These are the residue which are aligning with my sequence, and based on these scores, you can identify that okay, the total alignment score will be this, and because this is a good alignment score as compared to a bad alignment score, this is a better homologue as compared to the other homologue. Okay, I hope it makes some sense. Okay, now means let me just quickly take you through BLAST also, so that we don't leave that as such. Okay, so just give me 1 minute. Let me open a small presentation. Okay, just to show you the BLAST part. So I hope you are able to see this. Okay, let me see if I can view in a... Okay, you are able to see this now. F11. Okay. So basically, I mean, this is just a very general slide which have made. But let's say if you want to know about this particular sequence, that who will be the closest homologue of this particular sequence. Okay, so obviously you have several questions in your mind that okay, this is my sequence, and maybe it is an unknown sequence. So what is the sequence? What, from where it is coming from? What type of domains it has, and all these kind of questions you will have. So the overall idea here is that you start with a query. Okay, you start with a sequence which you have already identified either from here, either from your next generation sequencing data, or maybe from somewhere else. But now you have that particular sequence. Let's start with here. Okay, so you divide your sequence into different type of words. Okay, so like word is made up of a minimum number of sequences which is predetermined, and you can even change. So let's say for protein part, a word length should be minimum 3. Okay, so if a word length is 3, then there can be around 9 words generated from this 11 amino acid sequence. Okay, so like YAN, ANC, NCL, CLE, LEH, something like that. Okay, so basically the formula for this is L minus W plus 1. Okay, so based on L minus W plus 1, you will be able to get the total number of words. Now, once you have total number of words, what you will do? You will try to identify the possible combinations. Okay, that how many combinations will be showing a maximum alignment score. Okay, so LEH will show the maximum alignment score like 17. If LEH will come with LKH, it will show only 13. Means you can use that scoring matrix and you will find it out. Okay, so L to L, let's say, where is my L to L? So L to L is 4. Okay, E to E is 5. 5, 4, H. Where is H? H to H is 8. Okay, so how much is this? Okay, so it becomes 17, right? So it is 17 here. 8 plus 5 plus 8 plus 5 plus 4. Okay, so in that way, similarly, LKH will be 13 and all these things. So now we got like these 4. This is our score threshold, let's say 10. So we selected like LEH, LKH, CH, and QH. Okay, once we select these, now we will try to find out that where these hits are present in our database sequence. Okay, so we'll try to find them in our database sequences. And again, once we find them in exactly our database sequences, what we will do, we will start extending in both directions. Okay, we'll start identifying that who is the next hit and what it is matching with. So let's say QEH is matching with QEH, then K is matching with K, M with R, G with G, S with W, and in that way you will keep adding their score. Some of them will be negative, some of them will be positive, and based on that, the total score you will keep calculating. Once you will keep calculating in both directions, as you can see here, okay, you will get like scores for each one of them. So let's say for one you get 55, another one 51, another one 45, 39. Okay, then you can sort them by score and say that okay, this particular sequence or this particular homologue has the maximum score, and that's why it will be like the best homologue. Okay, so in that way you identify that okay, this will be our best homologue. So now this thing you will run like multiple times in a dynamic programming module, and then means you will be able to see the result in a very quick manner. Okay, so dynamic programming will help like more in terms of getting the result very quickly. Okay, so this is like kind of a broad strategy for like the BLAST algorithm. Okay, now there is another word which is e-value. Okay, like in the BLAST part you will see it, that e-value, that how much is the e-value, and the e-value means that what are the chances of getting a hit just by chance alone. Okay, so if because these are computational methods, and there can be a possibility that some hits you are getting just by random chance. So to remove that issue, they have created this e-value, expect value, and if expect value is like really low, then it means that's a good hit and a confident hit of this particular this particular tool. Okay, so I hope this makes some sense here. Okay, so based on these different strategies, especially the BLAST part, now this homology analysis can be used in different studies. Okay, so when we do like computational, like comparative genomics of 100 organisms or maybe 200 organisms, so the idea is that you have, let's say, in open access data, there are 100 genomes available for Vibrio or 100 genomes available for E. coli. Then you can use those E. coli strains, all of them, and study them all together and find out that, okay, if there is any biological question, you can answer based on that analysis. Okay, so let's say sometimes it's a possibility that one of the organism has a genomic island, okay, like this. Okay, but this genomic island is absent in others. As you can see, it's not there, right? So these this type of studies are known as synthetic studies. Synthetic studies means that you try to compare multiple genomes all together one by one or in a group, and find out that where the, like, how the order of the genes is maintained or not. Okay, whether the order of the genes is maintained or not. If it is maintained, it means they are synthetic. If it is not maintained, it means something unique has happened in that particular genome. So let's say here, this is very different as compared to others. So it means that this particular organism has genomic island, others don't have. So maybe others lost it, or a possibility is that this particular organism gained this type of genomic island, right? So based on this, we try to use this type of homology studies in synthetic studies. Similarly, we can use, so means I hope you remember I talked about complete genomes, draft genomes like contigs, scaffolds, and chromosome. Okay, so contigs are unordered, unordered. Scaffolds are ordered. Okay, so if you have a reference genome, which is kind of complete genome, and then what you can do, you can arrange these contigs. That okay, this contig should come here, this contig should come here, and this contig should come here. So this is known as genome arrangement, genome ordering. Okay, ordering of contigs. So there are like some well-defined tools like MOV. So this is a tool, MAUV. This is U and E. Okay, MOV. So this MOV can help you organize your contigs with a reference genome. Okay, so if this is your reference genome, this is your contig, you can see that this, it is saying that this is showing maximum homology with this is showing more homology with this. So basically, this two contigs or these two genomic segments should be swapped. Okay, so you can do this kind of studies also. Another very important and very pertinent field is like pan-genome studies. So in pan-genome, what we try to do, like when we have multiple genomes to study, let's say again 100, 10, 20, 50, then we want to understand that, okay, let's say I'm working with E. coli, 10 E. coli genomes, then out of those 10 E. coli genomes, remember, all of them belongs to E. coli. So they are different strains. So ideally, they should be very much similar to each other. But even then, there can be some genes which will be absent in a second organism, or which will be unique in third organism only. So in that case, we want to understand that how the gene content is different between different organisms. So in that case, we do this kind of pangenome studies, which says, or using which you can identify that which is the core gene, which means that they are common in all the strains, which is unique genes, which means which are uniquely present in that particular organism, and which are dispensable genes. Dispensable genes means that they are important for two organisms, but not for the rest of the 8. Or they are important for maybe 7 organisms and maybe not for the rest of the 3. Okay, so in that way, we define like these 3 different modules here: core, accessory, and unique. And as you can see here, so if more closer organisms will have more number of core genes and less number of dispensable and accessory genes. But more distinctly related organisms will have less number of core genes and more number of dispensable and unique genes. Okay, so that's how you define these things. Okay, so another thing here is, okay, so this is like more about like, so you can use these homology studies in pangenome also. Another very important thing is obviously the phylogeny studies, which we keep doing, and I'm sure like there have been some sessions on phylogeny earlier. Okay, so I will not go too much into detail, but the idea is again here based on homology, that if there is a protein of your interest or nuclear gene sequence of your interest, you can identify the homologous sequence, you can identify, align them all, and find like phylogeny parameters and all, and then generate the phylogeny. Once you have the phylogeny, you can see their relationships. Okay. I just want to pinpoint like two main points here. One is like gene tree, another is species tree. So remember, housekeeping gene or a concatenated housekeeping genes tree are more of a species tree, which means that you are trying to understand the location or the relatedness of organism. So when you are trying to identify the relatedness of organism, that is a species tree, and they are based on speciation events. But when you are trying to understand the evolution of a one particular protein, they are known as gene tree, and gene trees are more based on mutation events. Okay, if mutation events and speciation events are occurring at the same time, then both of these tree will be same, but they don't occur in the same time, and that's why both of these tree are very different. Okay, species tree is the real tree which says about the organism, and protein tree is on will only talk about that particular protein. Okay, a protein may evolve at a different rate as compared to the organism. Okay, so just remember these two terms. And yes, I think with this, I will skip all these slides because of running late also. Okay, but this was more of like our work, which I wanted to discuss. But let's go towards the conclusion. And quickly, I want to convey that that future of bioinformatics is really bright. Okay, so I really want to focus here on obviously learning and getting trained in bioinformatics if possible, because bioinformatics is going to stay, and like it's one important field nowadays. There is a lot of data, exponential increase of biological information. Okay, so biological information is increasing extensively, in especially in computational domain. Multiple layers of information is coming, like omics, genomics, metagenomics, transcriptomics, ionomics, so many. Okay, and now then there will be more requirement of integration of these. Obviously high throughput technologies are way cheap now, so again it will increase the amount. More computational resources are available, which will again, people will be able to analyze more, and more analysis means more data availability. Okay, nowadays obviously there is like more awareness about bioinformatics standards, like good bioinformatics practice. Okay, so and obviously there is a huge need for micro and macro analysis, and obviously requirement for better algorithms. So based on all these things, I can definitely say, and everyone will agree with that, that the future of bioinformatics is really bright. And one another thing which is sometimes we keep forgetting, that bioinformatics, computational biology is an integral part of biology. Okay, and molecular biology, obviously it is moving very fast, but it cannot move this fast without computational biology. So computational biology is really an integral part of biology, and computational biologists are also biologists, which we are using just different tool. Okay, and it lets you see a very big picture, so it can help the molecular biologist immensely. So once you start learning it, it will really help you in a long run. Okay, and before I leave, I just want to say that even computational biology is not just running blast and doing the and seeing what you are getting. Okay, you need to have a plan, you need to have some kind of biological question, you need to answer some kind of biology in an output. So similar to experimental biology, understand your input data, understand your source from where you are getting it. Is it a good source, or is it really quality or curated source? Understand the methodology which you are following. Okay, if you are not if you are just randomly following the different type of methodology, you will not be able to get like the right answer. Okay, have a plan to get from point A to point B. Understand your equipment, similar to experimental biology. Understand your equipment, software, program, tool, and its methodology. Don't just run blast immediately. First try to understand what is blast, what are different parameters, how they will help me, all these kind of questions, which type of database I should select. Only then you should do these things. Okay, be critical and understand potential source of error. Okay, when you don't get your gel as you want, you try to see that what was the error, where might be the error coming from, right? Similarly, try to identify the error. Don't believe what you are seeing immediately. Okay, think about it twice. Okay, try to interpret your results. And again, reproducibility is also very important, similar to experimental biology. Don't just what you are getting in the first attempt, don't just make it a result. Okay, try to do it like 3 times. 4 times and see that if you are getting the right answer again and again. Okay, so obviously computational biology is a little bit cooler than experimental biology, and like I'm sure you know now. Okay, computational biology are like more chill people, good people, similar to experimental biology. Okay, but yeah, we have some good benefits of working on computers instead of in lab where we have to follow like very restricted practices. Okay, and with this, I just want to acknowledge like all the people and especially Dr. Shinoy who invited me for this training program. Or means I don't know if it was like really training as such, but my idea was that in 2 hours we cannot provide like real training on computational tools and everything. And when the topic is like this broad, it becomes a little bit problematic. And I also want to say that we are hiring. So in case you are a master's pass out with a fellowship, so I just want to pinpoint especially that the person should have fellowship. Only then we can take that person as a PhD student and in case you are willing to write postdoc fellowships, again, if you have computational biology experience only those people we are considering right now. So if you have computational biology background, or if someone of your friends or someone has computational biology background, let them know. So I also want to pinpoint that on my website, I have created like a Google form for showing interest. So the idea is like, if you will show interest, please show interest only via that Google form, because tracking email is a complicated task for everyone. And I can easily see the Google sheet and find out that, who has applied and everything. Okay. So yeah, thank you very much. And thank you very much, Dr. Shinoy. Yeah.
Thank you, sir. Now the topic is open for discussion. Anybody from the participants can ask their questions.
Dr. Shinoy has typed a question in the chat box.
Yeah, means that is a very good point, sir, because in prokaryote, at least we have, and on average, if we talk about like E. coli type of system, we have 10 to 15% hypothetical proteins. But if we talk about some less explored system, we have 30 to 40% hypothetical protein. And it is a big issue because out of that 30-40%, at least 20% doesn't show any homologue in any other organism. Even if it shows homology, it shows with the hypothetical protein. So in that case, it is kind of a chicken and egg problem. Okay, but yes, we need to explore more, and obviously molecular biologists need to explore more in terms of function, because as I said, the function which we are finding because some molecular biologists worked on that earlier and suggested that, okay, this protein should have this function. And as a computational biologist, we try to find homology, and based on homology, we try to predict the function. So in that case, it is kind of a on loop situation. So we are not able to find, but it means that because molecular biology has not found it, so we are not able to find it. In eukaryotes, yes, there are way more hypothetical proteins. Means I'm not really aware about the number as such, but they have more.
Okay. Anybody else from the audience, participants? Someone is asking.
Point, but for everybody, this is an information that the session has been recorded and it will be available on YouTube on MycoAsia channel in some time after editing, of course. So PowerPoint will not be shared, but the session will be available online. And thank you so much, Gaurav, for a very informative session. I myself learned a lot of basics. Thank you so much. And over to Romana for vote of thanks.
Thank you, ma'am. Thank you, Gaurav sir, for your valuable knowledge regarding genomic sequencing and highlighting on the areas like first generation, second generation, third generation sequencing, and especially highlighting that differences between different areas such as metagenomics, bioinformatics, and so on. Thank you very much, sir, for such a valuable, knowledgeable session. And I also thank Shinoy, sir, for organizing this workshop. And I also thank MycoAsia team, Dr. Gunjan Sharma ma'am, Dr. Anandita, and Manohar for their kind cooperation throughout this workshop. Thank you, everyone.
Thank you very much. And yeah, stay connected, guys.
Have a good day. Thank you, everyone.
Bye.
Bye.